Alright, so now that we've covered the basics of terminology, homeostasis, cells and connective tissue, we're going to move on to the various body systems. The first system that we will cover will be the skeletal system, and we'll start today by looking at the bone anatomy. So the skeletal system includes all of the bones and the joints within the body, and each bone itself is a complex living structure. The skeleton acts as a bit of a scaffold by providing support and protection for the rest of the body. The skeletal system provides an attachment point for our muscles to attach so we can actually move our body. New blood cells are produced in the red bone marrow that sits inside some of our bones. And bones can also act as a storage site for things like calcium, for iron, and energy in this form of fat. So we will cover the skeletal system in 2 lectures, focusing in this lecture on bone anatomy, and then the following lecture will be on bone physiology. So today we have 5 learning objectives to cover, starting with being able to list and describe the functions of bone. So firstly, the skeleton or bone serves as the structural framework of the body by supporting the soft tissues and providing attachment points for the tendons of skeletal muscle. So without this structural support of bones, we would not be the upright, solid creatures that we are. Secondly, the skeleton and the bones act to protect our most important internal organs, and particularly those organs that are soft and don't have their own protective mechanisms. So for example, our skull will protect the brain, and our ribs protect the heart and the lungs. Thirdly, the attachment of skeletal muscle to bone is what allows us to move. So when a muscle contracts, it will shorten and it pulls on one or more bones to produce a movement at that joint. So we need our bones for our muscles to actually function. Fourth, bone tissue plays a role in the homeostasis of minerals. So we have large amounts of calcium and phosphorus that are stored within the bone, and this is actually what gives bone its hardness. About 99% of all of the calcium in the body is stored in our bones. So what happens is when we have low levels of these different minerals within our blood, the body will actually break down the bone to release those minerals back into the blood. Similarly, when the concentration of these minerals is high in the blood, the excess will be taken out and stored in the extracellular matrix of bone tissue. Number 5, within certain bones, red bone marrow will sit, and our red bone marrow produces our red blood cells, our white blood cells, and our platelets. And so our platelets are the cells that help our blood clot. Red bone marrow is found in the developing bones of a fetus, as well as some adult bones, like our long bones, our ribs, our sternum, our hip, our humerus and femur. And lastly, we also have yellow bone marrow, which is found within the medullary cavity of long bone. So that's the kind of inner middle section of a long bone. And yellow bone marrow is made up largely of stored triglycerides, so of stored fat. And so we can actually use this as a bit of an energy reserve. So now that we know what bones do, learning objective number 2 is to compare the different types of bones and provide an example of each. So all the bones in the body can be classified into one of 5 different types of bone. I'm going to give you a few examples here, but please don't stress about writing any of this down. We will cover the definitions of each type of bone on the next few slides. So we have long bones. Long bones are long, and they include things like your humerus, the radius and ulna, which are our lower arm bones, our femur, and our tibia and our fibula. We also have long bones in our fingers, which, although are not particularly big bones, they are quite long. We have short bones, and similar to the long bones, it's not necessarily referring to the overall size of the bone, but its relative length compared to its width, so it's relatively short. We find these in the bones of our wrists and in the bones of our ankle or our foot. We have flat bones, which are flat. So examples are the sternum and the bones of our skull. We have irregular bones, and irregular bones are typically complex, unusual shapes and bones that don't fit into any of the other categories. So irregular bones include bones like our vertebra, which make up our vertebral column, as well as the ethmoid bone in the brain or in the skull. Lastly, we have a sesamoid bone, and a sesamoid bone is a bone that is embedded within a tendon. So the most obvious example of a sesamoid bone is our patella, which is also known as our kneecap. And the patella sits within that patella or the quadriceps tendon. We also have sesamoid bones on the balls of our feet, and we actually have some really great models in the dry lab that show these really well. So make sure you have a bit of a look at those when you get the chance. So looking at how we specifically define these different types of bones, so short bones are about as long as they are wide. They provide stability and a little bit of movement, but not a lot. And the examples include things like the carpals in the wrist, so those small kind of rectangular shaped wrist bones, as well as the tarsals in the ankle. Our long bones are longer than they are wide, so they include the femur, which is the longest bone in the whole body, as well as the relatively much shorter finger bones, because they are much longer than they are wide. Long bones function to support the weight of the body as well as facilitate movement. They're long levers to facilitate movement. Long bones are also mostly located in what we call the appendicular skeleton. So this is your appendages, so your arms and your legs. We also then have the axial skeleton, which is more the midline of the body. So that's your skull, your vertebral column, and your ribs. Some examples of long bones include the lower limbs, so the tibia, the fibula, metatarsals, and the phalanges in your feet. And bones in the hands and the arms, so the humerus, the radius, and the ulna, your metacarpals, and the phalanges in your fingers. Flat bones are flat and relatively thin. There are flat bones in the skull, so our occipital, our parietal, and our frontal bones, as well as the thoracic cage, so your sternum is a flat bone, your ribs are flat bones. And the function of these bones is to protect our internal organs. So the skull protects the brain, the ribs protect the heart and the lungs. Now, although flat bones are called flat bones, and they are somewhat flattened, they can be slightly curved. So don't think of it as like a flat piece of paper. Just think of it as a thin type bone. Aside from protection, some flat bones also provide a large area of attachment for muscles. So our scapula is another example of a flat bone, and we have lots of muscles around the shoulder joint that attach directly onto the tissue of the flat bone. Our irregular bones will vary in shape and structure, and they typically just do not fit into any of the other categories. They often have a fairly complex shape, and many of them also help protect our internal organs. So again, the most obvious example is probably our vertebra. We've looked at those, they're quite interesting shapes, and they protect the spinal cord, which runs through the middle of all those vertebrae. Lastly, we have the sesamoid bones, which are the bones embedded in tendons. These are small, round bones, and they're commonly found in the tendons of hands, knees, and feet. And sesamoid bones work to protect tendons from stress and wear. So in joints that are used a lot, like our hands, our knees, and our feet, they help protect the tendons somewhat. So moving on to learning objective number 3, we are now going to look in more detail at the anatomy of a long bone, so just that one particular type. So you will need to be able to describe the structure and the function of each part of a long bone. So this here is an illustration of a long bone. Although obviously a drawing, it is very similar to what you'll see in a cadaver. And you will get the chance to look at a dissected long bone in class. You can also just go to your butchers and ask them to cut a long bone in half. It's typically the marrow bones that people will give their dogs. And so we're going to talk through each of these structures labelled here. And as I'm speaking, it might be a nice idea to pop down a really brief function for each structure that I mention. So we'll start with the diaphysis. And the diaphysis is just this central part of the bone. Now I know I said you should have a function for each part, but the diaphysis really doesn't have a function, though you could say that it contains the medullary cavity. We then have our two epiphyses. So remember, epi means on top of. So epi, it's on top of the diaphysis. So here and here. Our distal epiphysis is going to be further away from a point of attachment. So this here is a humerus bone or a upper arm bone. Our shoulder joint would be here. Our elbow joint would be here. So this is more distal to the point of attachment, and this is more proximal to the point of attachment. So proximal epiphysis, distal epiphysis. If we gave you an image to label or a particular epiphysis to identify, we will give you a bone that is really easy to work out what it is and which end is proximal and which end is distal. Our epiphyses are where another bone will articulate. Speaking of articulating, we have articular cartilage, which is that hyaline cartilage, that really smooth, clear cartilage on the ends of our epiphyses. The function of articular cartilage is to make a really nice, smooth surface for the two bones to rub up against each other. We don't want friction in those joints. We have our periosteum. So our peri means around, so it's the layer that wraps all the way around our bone. It's the very outer layer of all our bones. We then have the medullary cavity, and so this is the cavity or the hole that sits along the diaphysis. And within our medullary cavity of a long bone, we will have our yellow bone marrow. So remember, our yellow bone marrow is mostly made up of those stored triglycerides, and we can use that energy when required. We then have the end osteum, so end means in, within. So now this is the layer that surrounds the medullary cavity. It lines the inside of the bone. We have compact bone, which we're going to talk about in a couple of learning objectives times. We have two different types of bone. We have compact bone and spongy bone, but compact bone is this hard external layer of the bone. It's nice and compact and thick and acts as a protective function for the bone. We have the epiphyseal line, which is this white line here that kind of connects our diaphysis to our epiphysis. And the epiphyseal line is where we have growth of a long bone. So as we grow from children to adults and even further if you're lucky, our bones will grow up this way. This is where that growth occurs, and we'll talk in more detail about how this actually happens in the lecture tomorrow or the next lecture. Within the epiphysis, we have red bone marrow. So red bone marrow produces our red blood cells, our white blood cells, and our platelets. We often get our red bone marrow and our yellow bone marrow confused in what they do. Lots of students will tell me that red bone marrow produces red blood cells and yellow bone marrow produces white blood cells. No, red bone marrow produces all our blood cells. We don't have yellow. Within the medullary cavity where we have our yellow bone marrow, that's where we have that stored triglyceride. Spongy bone is the other type of bone that we have. So we have the compact bone and the spongy bone. It's a bit more spongy, it's a bit more light. It lightens the end of the bone, and our red bone marrow sits between the pieces of spongy bone. And then we have our articular cartilage again. So this is another point of articulation where the humerus will interact with the shoulder joint, the bones of the shoulder. So we have that nice smooth surface for those bones to glide over each other, so there's no friction in that joint. Alright, so we've looked at the gross or the large anatomy of a bone. Now we're going to take a bit of a look at the microscopic anatomy and what cells actually make up bone. So I want you to be able to compare the functions of osteoblasts, osteocytes, and osteoclasts. So bone is a tissue in which the extracellular matrix has been hardened to accommodate its supportive function, so to make it able to hold up the body. The fundamental components of bone, like all connective tissues, are cells and the extracellular matrix. And remember, the extracellular matrix is made up of ground substance and those different types of fibers. The extracellular matrix of bone contains mostly collagen fibers. So remember, our collagen fibers are the really strong, force-resistant fibers embedded in a mineralized ground substance. So the ground substance contains hydroxyapatite, which is basically just a form of calcium and phosphorus, but it's what gives bone its hardness. So I'm not going to test you on this, but just know that if you see that, it's stored calcium and phosphorus, and it's what gives bone its hardness. Now, both of these components of the extracellular matrix are really important for giving bones its strong property. If we didn't have the collagen fibers, bones would be really brittle and they would shatter quite easily. And if we didn't have the calcium and the phosphorus, our bones would just flex and they'd provide very little support, particularly when the muscles pull on them to create that movement. Now, within bone tissue, there are 4 different types of those specialized bone connective tissue cells. We have osteoprogenitor cells, we have osteoblasts, osteocytes, and osteoclasts. So looking at those 4 cells more closely, and this figure is showing basically the lifespan of a bone cell. So remember, all connective tissue, including the bone connective tissue, is derived from mesenchyme. So our osteoprogenitor cells are our earliest specialist bone cell, and these are derived from that mesenchyme. Looking at the name of that cell, so ost means bone, o is that combining word, progenitor means where something originates from. So an osteoprogenitor cell is where bone originates from. Osteoprogenitor cells will then develop into osteoblasts, and we've talked a little bit about osteoblasts already. So ost means bone and blast is an immature bone cell. So osteoblasts are our functional immature bone cells. Our osteoblasts will then develop into osteocytes. So again, ost, bone, o, combining word, cyte is a mature cell. So it's our mature bone cells. And then we have osteoclasts. And osteoclasts are slightly different because they are derived from white blood cells. But again, looking at that name, ost is bone, o is a combining word, and clast means to break down. So the function of this cell, which we will talk about in a few slides' time, should be somewhat obvious. So looking at each of those cells in turn, our earliest specialist bone cell is our osteoprogenitor cell. So the cell from which all bone cells will derive from. Our osteoprogenitor cells are undifferentiated bone cells, which are derived from that mesenchyme. Osteoprogenitor cells are located along the inner portion of the periosteum, so the inner layer that surrounds the whole outside of the bone. The endosteum, so the inner layer of the bone that surrounds that medullary cavity, as well as within the canals within bone that contain blood vessels. So we need little tunnels within our bone so that blood vessels can get into the bone, osteoprogenitor cells will line those little tunnels. Now, osteoprogenitor cells are the only bone cells to undergo cell division. So they will split and divide, and the resulting cells will develop into our osteoblasts. So our osteoblasts are the immature but our functional bone cell. They are the cells that build bone. So osteoblast, build bone. Osteoblasts secrete what we call osteoid, and osteoid is made up of the collagen fibers as well as the other materials required to make that extracellular matrix, so required to make that ground substance. Osteoid forms a matrix that is able to bind calcium and phosphorus. When calcium and phosphorus bind to that extracellular matrix, that matrix then becomes hard in a process called calcification. So we take the calcium and the phosphorus out of the blood, it goes into that ground substance, makes it hard, and this is what we call calcification. When the osteoblasts secrete this osteoid, it secretes it all around that osteoblast. As the osteoblast becomes trapped within the osteoid, it turns into our osteocyte. So it's secreting all this osteoid, it eventually becomes trapped within the osteoid, and that's when it turns into our osteocyte. The space within which the osteocyte is trapped is what we call the lacunae. So our osteocyte is an osteoblast that has become trapped in the osteoid or the calcified extracellular matrix. It's a bit of a mouthful that it secretes. So it forms itself a little cave. As I said before, the space that an osteocyte fits within or sits within is called a lacunae. Because they are now trapped within that lacunae, osteocytes will communicate with one another through long processes. So these long processes that extend out from the osteocyte within caniculi. So caniculi are just little tunnels that will connect the different osteocytes or connect the different lacunae. These caniculi are used for the transport of nutrients and wastes, as the primary function of an osteocyte is to maintain bone metabolism. So take nutrients to the bone cell and take the waste products away. So it's keeping the bone tissue alive. Lastly, we have our osteoclasts, and these are actually really big bone cells. They're derived from white blood cells, and they originate when as many as 50 monocytes, which is a particular type of white blood cell, fuses together. Our osteoclasts will migrate from bone marrow into the bone tissue, and they're concentrated in the endosteum of a bone, so the lining of that medullary cavity on the inside of the bone. Osteoclasts have this kind of ruffled border, and that ruffled border is what adheres to the bone extracellular matrix. When it attaches to that extracellular matrix, it will then secrete a number of different digestive enzymes and digestive acids. And this will break down the bone in a particular way. Remember, clast means to break down. This process of the osteoclasts breaking down that extracellular matrix is what we call bone reabsorption. When an osteoclast breaks down that extracellular matrix, it takes the stored calcium and the phosphate and puts it back into the blood so that it can be used in different parts of the body. So that was a lot. So I thought I'd give you a bit of a written summary of the last few slides. So osteoprogenitor cells are derived from mesenchyme. Remember, mesenchyme is that tissue from which all connective tissues originate. Osteoprogenitor cells will divide. They're the only bone cell that can divide, and then differentiate or develop into our osteoblast. Osteoblasts secrete osteoid, which is that unmineralized bone matrix. Osteoid is mineralized by the deposition of calcium hydroxyapatite, which is just the deposition or the binding of calcium and phosphorus into that extracellular matrix, and it makes that tissue hard. Our osteoblasts then become trapped in the lacunae, and now we call them osteocytes. Our osteocytes regulate bone metabolism, so they're involved in bringing nutrients to the cells, taking away the waste. And then when stimulated, and in the next section we'll talk about what actually stimulates osteoclasts, our osteoclasts will migrate from the bone marrow to the bone and complete that breaking down process, that resorption. So our last learning objective is to describe the histology or the makeup of compact and spongy bone. So if you remember back to the last lecture when we looked at all the different classifications of connective tissue, and I said that there were two different types of bone. Well, these are it. We have compact bone and we have spongy bone. So as the name implies, these two different types of bones differ in density, or how tightly packed the tissue is together. So on the outside of the bone, so you can see it here, and in a really thin layer kind of along the top of the epiphysis here, we have our compact bone. And you can see it's nice and compact. You can't see any spaces in that compact bone. A little bit within the medullary cavity, but then much more in this epiphysis, we have our spongy bone. And again, you can see it's more spongy. There's a lot more air-filled spaces within that bone tissue. So compact bone, which is also sometimes referred to as cortical bone. So if your textbook refers to cortical bone, it's just talking to compact bone. And it's a dense bone in which the extracellular matrix is solidly filled with the collagen fibers and that calcified osteoid. It leaves only tiny little spaces, which are the lacunae, to contain the osteocytes or the bone cells. So you can see in this image here, the little black dots are our lacunae and the osteocytes sitting within them. Apart from those spaces, there's basically no space in between the collagen fibers and the osteoid. So compact bone makes up about 80% of the human skeleton, and it's found in the hard external layer of all bones. So it surrounds the very outside of all bones. Because of its structure, compact bone provides strength, protection, and support to bone tissue. Compact bone is made up of what we call osteons, and I'll show you those in the next slide. But they're packed in nice and tightly next to each other, so you're not having much dead space in this bone tissue. So if we zoom in to see what compact bone looks like under a microscope, you'll see that it's made up of repeating units of osteons. So this circle part here is what we call an osteon. An osteon is kind of like a pillar of bone, and it's made up of lots and lots of rings of what we call concentric lamellae. So see how there's like a ring of bone here, then a ring of bone around that, and then a ring of bone around that, and then a ring of bone around that. Each of these rings is concentric lamellae, so just circular lamellae, so circular bone tissue. All of these together makes what we call an osteon. And so we say compact bone is made up of lots and lots of osteons. So walking a little bit more through this diagram, if we imagine this is, say, the just a section here of the diaphysis of a bone. So we're going to have the compact bone on the outside, and then you can see a little bit of that spongy bone on the inside. We have the concentric lamellae, which are those individual circular layers of bone. All of those together form what we call the osteon. We then have a central canal. So that runs down the middle of an osteon. And that central canal will house the blood vessels for the bone, as well as any nerves. So it runs down the center of each, you can see here and here, each osteon. We then also have what we call a perforating canal. Some textbooks will call this Volkmann's canal. And what that does is it actually connects the different central canals as well as connects it to the periosteum. So for these all to communicate, we have these perforating or these Volkmann's canals. What else I wanted to point out here is this periosteum. So this white layer here is that periosteum. It's the layer that surrounds all of our bone. And then here we've got a bit of a close-up look at that osteocyte. So in the middle here is our osteocyte. That is our mature bone cell, the bone cell that's responsible for maintaining bone metabolism. The space within that osteocyte sits is called the lacunae. So all of this, I guess, yellowy beige colour is secreted osteoid. It's trapped itself within it, so now this is an osteocyte. The space within which it sits is the lacunae. Remember that our osteocytes have these processes that extend out from the osteocyte, and that's so the different osteocytes can communicate with one another. Otherwise they'd just be isolated and they would die. The little tunnels with which these processes extend so that they can communicate with another osteocyte is called a caniculi. So I've introduced a lot of new terms for you there and kind of interesting, strange words that you've probably never heard before. So I wanted to pop up a bit of a definition of each of these words so that you can refer back to this slide as you're going through the lecture again, if you're revising or you just need to go over it again. I'll also pop all these words up on Canvas just so you have that as a resource there. But our lamellae are those concentric rings of calcified extracellular matrix. So those single rings within an osteon is called our lamellae. We then have an osteon. So that's a compact cylinder of concentric lamellae. So when all of those rings of lamellae are joined together, it forms that pillar of bone. That's what we call an osteon. We have the central canal, and so that's the tunnel basically in the center of the osteon that houses the blood vessel, so the nerve and the artery and the vein, as well as any nerves. We have the perforating or the Volkmann's, so I think that's Volkmann canal, which connects the central canals and the periosteum. So it connects basically the blood vessels to the osteons to each other. And then we have the caniculi, which connects the lacunae and the central canal. So the caniculi are the little tunnels where the processes of the osteocytes can communicate with one another. It also needs to communicate with the central canal so that we have some blood source going to those osteocytes. So the other type of bone we have is our spongy bone. And spongy bone is also referred to as cancellous bone. So again, some particularly older textbooks will refer to it as cancellous bone. Spongy bone reduces the density of the bone. It makes it a lot lighter, but it also allows the ends of long bones to compress to a degree. So when they've got things bearing down on them and forces coming in lots of directions, it actually allows it to compress somewhat because it's not as dense. Spongy bone is prominent in areas of bones that are not heavily stressed or where stresses arrive from many different directions. So for example, the epiphyses of bone, so each end of the bone, think of the head of the femur or the neck of the femur as an example, they're going to be stressed in lots of different directions. They've got muscles attaching in all different directions, so they're going to be stressed in a lot of different ways. Now, in spongy bone, instead of bone tissue being arranged in those bone pillars that we call osteons, they're arranged in trabeculae. So instead of those rings of lamellae being arranged in nice tight circles, they form more of an open kind of mesh network with lots of space in between. So if you look at this image here, the pink is the stained bone trabeculae, which are lamellae, and then the white is all the space in between. And in this case, this is where some yellow bone marrow sits. It's also where a lot of red bone marrow will sit. So it's not nice and tightly packed like our compact bone. So looking at the spongy bone up close, you can see that our trabeculae forms more of an open bone tissue than our osteons. So all this yellow is space between the trabeculae. Aside from not being as tightly packed as a compact bone, so having trabeculae instead of osteons, you can see that spongy bone is otherwise exactly the same as compact bones. So we have the same cells, the osteocytes, the osteoclasts, the osteoblasts. We have those rings of lamellae, but they form kind of crossbars with lots of space in between instead of all just being nicely packed circles that are all closely packed in together. So this is just an image of actual spongy bone. It's a photo, and you can see how much more of a open network it is compared to that compact bone. So this space in here is where all of your red bone marrow would sit. So again, a few new terms introduced in the last few slides, so another lot of definitions. So we have our trabeculae, which is just our lamellae arranged as beams or crossbars in a loose 3D network. I should have also mentioned in the last slide that our trabeculae, although it appears quite an unorganized mess, the trabeculae are actually arranged and will remodel along lines of stress or compression to strengthen that particular part of bone. So in the next lecture, we'll talk about bone remodeling after an injury or after it has a particular stimulus, which is often, for example, weight training or force or compression on that bone. And it will remodel to have more trabeculae in a particular part of spongy bone where there's a lot of stress. The other term we introduced, which we have mentioned a few times, but is our red bone marrow. So this is found in between the trabeculae and it produces our blood cells. So the red blood cells, the white blood cells, and our clotting cells, the platelets. So that is it for bone anatomy. We will finish off the skeletal system in the next lecture when we look at bone physiology and how all these different structures actually function. So thanks again for listening, and I will see you next time.